Hello, dear students. In last lecture, we have discussed about the different types of microorganisms and their habitat. Now, we discuss about the different major characteristics that are used in the taxonomy for the classification of these microorganisms. Let's see one by one. It is a typical structure of bacteria. So, identification of an organism is the process of determining its species. Various characteristics of the organism are determined by the appropriate observation and test, and for this they are compared with the public description of the various species, and identification of microorganisms is done on the basis of morphological characteristics, physiological characteristics, immunological characteristics, their metabolic processes, etiological characteristics, their protein composition, their nucleic acid composition, hybridization process, nucleic acid sequences, either section is RNA or section is rDNA. So let's discuss one by one. What is meant by the morphological characteristics? Apart from other kinds of microorganisms, microbial characteristics, determination of morphological features usually require study individual cells of pure culture of microorganisms. They are very small in size, usually expressed in micrometer. Size, shape, and arrangement of cells determine the microscopic examination of stain smear. Examination of living organism in a hanging drop preparation is useful for the determining the motility of microorganisms. Uh, the organisms are differentiated on the basis of various staining reactions. Frequently used staining methods are gram stain, acid fast stain, flagella stain, capsule stain, pore staining, and negative staining. So these are the different staining techniques used for the differentiation of whether an organism is gram positive or gram negative, acid fast or gram acid negative, whether it contains flagella or not, whether they have capsule or not, whether they are endospore former or not. So these are the different characteristics, morphological characteristics which can differentiate different microorganisms in another. Gram-stain is a great value because organisms are classified as gram positive, gram negative. The staining reaction, however, vary with the composition medium, composition of the and age of the culture. So another feature is the cultural characteristics. It is nothing but a pure culture of organisms can be studied by growing them on a wide variety of culture media. Under optimum conditions, cultural conditions, organisms show characteristic types of growth, and such observations are useful in the identification of the organism. Growth characteristics are commonly observed on the following types of culture, like colony on solid media, that is plate culture, in liquid media, in on slant culture by using stabbing culture or in gelatin stab. Description of colonies on solid media should include size, shape, elevation, surface color, and appearance by reflected and transmitted light. So similar information can also be secured from agar slant and agar stab culture. So these are the cultural characteristics. Uh, Colony characteristics. So when this pure culture is grown on the different solid media, a typical growth of bacteria can appear with respect to the media which can be used for the uh, inoculation of the microorganism. So this is a typical growth of microorganism, and a single colony can be differentiated using with respect to their margin, color, elevation, texture, shape. So these are the different characteristics growth of microorganism. So it is again another feature. Bacterial growth pattern on plant and liquid media. So when they are grown on the liquid media, it is formed a typical type of colony. It is a simple uninoculated culture growth. When they form a growth on the surface of the growth, they are called as spherical formation. When they settle down at the bottom, they are sedimented. When they form uniform turbidity in the medium, they are turbidity. And they form flocule, they are flocule. Also on plant culture, they form fully formed, echinolate, beaded, effuse. So these are the different characteristics growth of microorganisms in solid media. Another is physiological characteristics of microorganisms. These characteristics are commonly used in the identification of organisms. as follows with respect to temperature range. That is, the organisms which are grew up to 0 to 20 degrees Celsius are called as saprophytes. The organisms which grew 20 to 45 degrees Celsius are called as mesophiles, while the thermophiles, which can grow up to 45 to 65 degrees Celsius, and the extreme condition that is 65 to 100 degrees Celsius, that organism are called as thermoduric. With respect to oxygen tolerance, strict aerobes requires oxygen for the growth, while strict anaerobes, their oxygen is lethal for the growth of this organism. Another is facultative anaerobes, that is, they does not require oxygen, but in the presence of oxygen, they can survive. Another is microaerophilic, they require 2 to 10% of oxygen of the growth. With respect to pH range, organisms are categorized into acidophiles, they are acid level, neutrophils, and alkalophiles. Pigment production, either water soluble or insoluble, tolerance and requirement of salt, and source of illumination, photosynthetic organisms. So these are the different physiological characteristics of microorganisms. With respect to immunological characteristics, certain chemical components of microbial cells are called as antigens. Every organism has its antigenic structure. Antigen characterization of microorganism has great practical importance. By using this, we can identify whether the serum or blood sample is uh, having the antigen of particular pathogen. If microbial cells enter the animal's body, the animal responds to their antigen by forming a specific blood serum protein called antibodies, which bind to the antigen. So this is nothing and treated by the antigen antibody reaction. Antibodies are highly specific for the antigen that induce their formation because different kinds of microorganisms have different types of antigens. Antibodies are widely used as tool for the rapid identification of particular kind of infection or microorganism. For example, if we take a typhoid bacterium antibody and mix with a suspension of unknown bacterial cells, if precipitation occurs, the Vidal test considered to be positive and considered as unknown sample is of typhoid bacteria or Vidal positive. So it is all about the different uh, physiological, cultural, morphological, immunological characteristics of microorganisms can be useful for the identification of microorganisms. So thank you.